Welcome, Mikael Eliasson from Roche.
So, great, thank you very much. Oh, and there are my slides too. So I'm so delighted to be here. It's been fascinating this morning. In fact, I already wanted to change my slides because I've learned so much about actually what we could do at Roche. So I was going to be upfront about this. I'm actually here to talk about my problems because I think you can really help me with them. So that's what my talk is going to be. So the source of my problems is this, the brain, the human brain. It's absolutely a fascinating thing. I mean, it's the most complex thing in the universe. I don't know, how many of you have seen a live brain? Oh, that's not that many. You really got to check it out because it's a remarkable thing. So, I went to medical school. So by the way, the surgeon talks this morning, amazing. So I trained in surgery. I don't know how many of you know this, but the way they select surgeons into surgery residencies is all theoretical. They don't test if you can tie a knot, if you can suture anything. They just ask you questions. It's like they would determine the champion of Wimbledon based upon, some theoretical construct. Here comes the ball. Should you hit it down the line, or should you hit it across court, or should you do a lob, right? But they actually don't test any practical skills at all, which is strange, right? And the other thing is that the mantra that the teaching hospitals, and I trained at Brigham and Johns Hopkins, is see one, do one, teach one. That's it. And you do that on patients. So that you're, like Walter was talking about, you're developing VR to find models where you actually train people to do this is great. We really need it. But anyway, back to my problem. So this is kind of my source of my problem. I remember the first time I saw a living brain. It's absolutely amazing. I was a medical student. When you go into the OR there, first you spend like 50 minutes washing your hands. And then you stand like this. Because the scrub nurse will tell you, don't touch anything with your hands because you're gloved in. So you're literally standing like this. It looks like you're praying. And you're like, and you're just listening to the beeping noises. And I remember this, right, because you look over to where they're doing the surgery and you can't see anything because the surgeons are all hovering over it. So all you see is this light blue clothing they're wearing. And then I remember Henry Brehm, who was the attending, he looked at me and said, hey, you want to see? And it, like kind of religious experience, the other surgeons just kind of stand aside, and there the brain was. And I remember walking up, being so afraid that I would trip and fall on it. So I kind of was walking without walking. So after a while, I was almost like leaning forward, like the ski jumpers when they take off to look at that. But the brain is an amazing thing. Like it pulsates, it's glistening. You see the veins and artery. But it really is just a gray lump. And that everything that we are is in that brain is just absolutely remarkable. So I'm kind of dedicated now to understand the brain better and help people with neurological disease. But I read this quote a couple of years ago, and I really like it. And it goes like this. If the brain is so simple that we can understand it, we would be too dumb to understand it. So maybe all this effort is futile, but we can always apply AI on it and we'll figure it out. So first, let me introduce myself. So I work at Roche, Genentech. It's a biotech. We're up the road here. It's about 2 hours by bike, by 3 hours by car because of traffic. We have several of my colleagues in here. We're about 4 or 5 here, so we have a large interest in neurotech and wearables in this space. But we are a biotech company. We're the largest biotech company in the world. And this is our neuroscience portfolio. I'm not going to talk about our neuroscience portfolio, but we have one of the biggest, if not the biggest neuroscience portfolio in the industry. Sadly, most pharma companies are exiting neuroscience because we're all going into oncology and rare diseases. So I'm going to talk about the problems we have in unlocking neuroscience. So here's the first problem. And it goes something like this story here, right? So it's a joke. So please laugh afterwards. But it's basically, you have a police officer who's walking down the street, it's dark, and you see this lamppost with a light. And under the light, this man is crawling around looking for something. So he goes up to him and says, hey, can I help you? And say, yeah, I'll drop my keys. Okay, well, let me help you find it. So they look at a little area of the light, and the police officer's like, well, that's weird, we can't find him because it's just right there. And he said, where did you lose them? Do you remember? And the man points into the alley. He said, why are you looking here when you drop them here? But this is where the light is. Now, this is a problem we have in science. We are stuck with improving things we can measure. We are not measuring the things we should be measuring because we can't. And that is the first thing we're trying to change at Roche. What does this mean in reality? What's a concrete example? So here's a very concrete example that you think we should have solved a long time ago. So each dot here, you can see it's kind of gray. It's a little bit hard to see. It's a day. So if you have multiple sclerosis, you typically are seen twice in a year. Oh, all right. So that's a blue dot. So you're seen by the doctor. And when you go to the doctor, you talk about how you're doing and we need to change the therapy. So if the red here is the disease activity, so a little bit more red, this means you have more disease. The doctor will kind of figure out, okay, so this is kind of based upon asking the patient how they've been doing, is the level of disease activity. So this is kind of what's captured in the records on the patient. Now we've been sort of developing tools now to measure continuous activity. So what we're trying to go from is this sense of measuring disease as an episodic, subjective, in the clinic measurement. Oh, you couldn't complete your purchase. We need to buy something else. To continuous, objective, kind of at-home measurements. So if you actually look at that, the actual picture of the disease is more like this. So that is different from this. Now, so the challenge we have at Roche, we develop drugs, right? You have endpoints, you measure disease activity. So if this is what it captures disease activity, but it's actually this, it's so inaccurate that you're going to get a difference between control group and treated group. It just doesn't work. So that's one challenge. So let me give you another example. So, good. 100 years ago, more than 100 years ago, this boy has Duchenne's muscular dystrophy. It's a disease of the muscles, and it's turned his muscles into fat. So this is not, he doesn't have big calves because he's strong, it's because there's fat. It doesn't work. He did something there called a 6-minute walking test, and now he does a stair climb. This is 1910, somewhere in Belgium. I'm showing you this because this is still the gold standard. If you want to develop a drug for Duchenne's muscular dystrophy, this is the same way, this is the endpoint you would bring to FDA to show that the drug is working or not. It hasn't changed over 100 years. There's problems with that. So what the company we've been working with called SysNav did, they put us, remember the panel here, this is a purpose-built sensor, ankle sensors that measure stride velocity. So it's not a consumable goods. It's built by this company that is also in the defense industry. But what they've been able to do, they measure the stride velocity of these boys. And they come up with this, so they know how fast they can walk. And they created this endpoint called 95% stride velocity. But anyway, that allowed you to check on the 6-minute walking test. So they basically tested it. See if I can get here we go. Can you see the red marker here? So they basically, on the x-axis, they call it kind of compliance. So this is how fast you can walk. And this is actually, so if the further you go down here, you're actually walking as fast as you can. And this just is test, retest, right? So the more higher you go up here, it basically means that your effort is not reproducible. So if you had a perfect test where the boy will basically do the best he can, and each time they will be down here. All of these ones are, they're not putting the same effort in. These kids here can walk much faster than they do. So why is this? Well, there are a couple of reasons for that. First of all, how exciting is to be told, okay, you have 6 minutes, you're going to walk back and forth here, and I'm going to time, you're going to walk as fast as you can. This is the motivation thing. The second thing, Dr. Servet, his practice is in Paris, France, right? So if you go to Paris, France, so he's going to see the kid on a Monday, the family will then fly in on a Friday. What do you do over the weekend in France? Well, you go sightseeing. You go to Euro Disney, you go to the Louvre, you go to the Eiffel Tower. So by the time Monday comes, they're tired. And that affects it, right? So the problem this noise adds means if we need to do a clinical trial using 6-minute walking test, we have to have something like, and this just shows the effect size, and the standard deviation is very high because you have very noise. And you need something like 112 kids for 12 months, so it becomes very expensive. It takes a very long time to do it. Now, if you use this 95 stride velocity on hand, the calculations they've done based on natural history studies that they show that the effect size is roughly the same. The standard deviation is much, much closer because the 95% stride velocity is very reproducible. That's why they picked it. And then their calculation, like, look, we wouldn't do a trial with just 14 subjects, but they basically said you can do 14 subjects in 6 months. If you look at the difference there, you're talking about years and hundreds of millions of dollars cheaper, right? So that's one way we try to create better tools. So what we have now at Roche, here we get it to slide change here. We built the platform using a smartphone. It's actually called Floodlight, which kind of helps with my, the lamppost thing. Now we have like a floodlight, so we can see light everywhere. But we basically used built this technology because, as you know, now smartphone is it's everywhere, and we use all kinds of different measurements, and it's basically now fully adopted into Roche. Sorry, the problem with the clicker here. So we have it in every one of our program at Roche. We're using these digital measurements. So it's something we have fully adopted. So the reason I come here was to kind of get ideas on what we can do next. And I love all of these sensors, sweat sensors and things you're showing here, measuring cortisol level and things like that. So that gives a lot of ideas. But in terms of smartphone measurements, we're kind of all in. And as much as any other company around here. The second thing is, so what's next? So the thing I'm very excited about at the moment I'm looking at and that we're having a conversation, and like Nathan's going to talk about it later, is EEG. And the thing that really piqued my interest and the opportunity here is that this is a picture of the retina of a human being. Now, who of you can tell me if this is male or female? Have you heard the Google story around this? So this is what I was told. It's actually someone from Stanford. I think she was an intern at Google, but basically given a task to come to work at Google. And this was told me by the team at Google that, hey, why don't you take all of these images, and we're talking about hundreds of thousands of images, and see if you can determine if this is male or female. So she went off and came back 3 days later. Yeah, you can. So she trained a neural net, probably convolutional neural network, recurrent neural network, to detect if you're male or female using this. I mean, it's a great way of using AI, right? Because it's all classified. Usually you know if an image, if it's male or female. So she trained it, and the AUC is like 0.97. Just like perfect. Now, but this taught me that there's no human being that can figure out if this is a male or female. And in fact, they've done a heat map of this, so they actually look at what the algorithms find the information. And this is like from everywhere. You look a little bit at vessels, you look a bit at the fundus. So there's not like, one little area you look at and it gives it away. But this neural net can take very complex information and gain insights from it. And that's why I have a lot of hope in EEG. I mean, EEGs are very, very complex to see. But I think hidden there's a lot of information that we can, if you use these neural nets and train them, we can pull out the information from it. In fact, there's just a paper published yesterday in Nature Biotechnology from a group here at Stanford, Amit Etkin's lab, that showed that they have found an EEG signature that predicts if you can respond to sertraline or not. So it's antidepressant therapy. So I see a lot of opportunity to find better biomarkers, understanding better what's going on in the brain from using EEG. So that's kind of my focus moving forward this year. Now, the second thing we're working on, the second problem is this. That's a pill or a capsule. And when and remember, I work for a biotech company. I don't work for a neurotech company. My job is to bring tech into biotech. But we are a drug company or biotech. We don't build technology. What I tell and what I say is this. Pills don't teach skills. Now, that is to say, there's no pill that will tell you how to speak another language, how to be better in social interaction. It's not even a pill that will teach you how to boil an egg. Now, why am I telling you this? Because we have programs where we try to go after behavioral disorders with pills. And the outcomes measurements on those is improvement of social interactions. So how's that going to work? For example, one program we work on, and the leader of that program, Kevin, is here. So I work with him on this. When we think about this, is in autism, if you give a drug to a kid with autism, how are you going to be better social interactions? So we're trying to find ways how we can augment the therapy, which probably will increase social drive. So you would want to have the interaction, but you won't know how you will have the interaction. So if you can combine that with digital therapeutics, it's something we look at. So one idea is, so how can we reimagine a company at Roche, how we treat illnesses? So one thing we've been working on, and hopefully we're planning to make some quite big announcements this year, so I can't tell you about it right now. But we're looking how we can use something like virtual reality to teach those social skills. And the principle around this is very simple. So I really like Walter's talk earlier today. But it's that, the second or third time you do something is easier than the first time, right? So if you have them interact in VR with avatars, to have small talk, or introduce themselves, we think that will translate into you feeling more comfortable doing this in the real world later. It's kind of a similar principle we put pilots in a flight simulator. So that's something we do. So we're trying to find a way of combining drug therapy, where it kind of creates the brain more responsive. We have a stronger drive to have social interactions, with something that teaches you how to do that social interaction. That's what we're doing right now. Then what I'm really excited about, we're not doing this at the moment, but where I think we really need to go, and that's why I really like the talk from Abbott. So remember, I went into neurosurgery 20 years ago because I wanted to get access to human brains, ethical access to human brains. But it also meant I needed to be a surgeon, and the technology just wasn't there to do what I wanted to do. So I left, and that's why I'm in industry now. But what I really like, what I find actually has a lot of potential, is deep brain stimulation. Right? So we need to move from model behavioral disorders where we try to treat by reducing some or increasing some level of a neurotransmitter like serotonin. This doesn't work that way. We have to actually drive circuitry, and stimulation is what can do that. I'm hoping increasingly we put these grids, like when I was a neurosurgeon, we put this grid on kids or adults that have intractable epilepsy to record where the focus of the epilepsy is, so just take out that area. But that also allows you to learn so much about the brain. I just learned from being visiting with a neurosurgery department that now they do that. You don't just put the scalpel electrodes on the surface of the brain, they actually do it indwelling. So they get three-dimensional recordings from the brain. And they're learning all kinds of things. And the patients who have that, they have it on for a week. So they sit, and you can do all kinds of interesting work and experiments looking at that. And it's a way to understand how the brain works. So I think there's a lot of opportunity here, and I love to hear the professor from Abbott around this, because DBS hasn't really moved on in the last 20 years. But I'm hoping we're starting to get at the tipping point. And I'm saying that for a couple of things that happen. One is analytics, right? So now we have these neural nets that can handle really complex information. That's the first thing. That's kind of already here. It used to be we didn't like a lot of data. Now we like a lot of data. Engineering is really picked up in this field. And it helps that Elon Musk formed Neuralink, right? It creates a lot of attention. So that's coming. In facility, social acceptance, right, is still not there to a large extent. Now, having said that, there's over 100,000 people who have been implanted with deep brain stimulation for Parkinson's disease. It's an established part of that therapy. But if you start moving into behavior disorders and things like that, there's still really not acceptance. So I hope that would happen at some point. So when I woke up this morning at 4 a.m., I was thinking about, what will happen for the kind of world to change? And I thought about it, and I started thinking about how was the world in 2000. So I created a slide to kind of see how kind of the world changes over time. So bear with me here. So in 2000, the kind of big problem everyone was worried about was Y2K. I don't know if you remember that, like all the computers were shut down whatever from 1999 to 2000. Turned out nothing happened. The movie that won the Academy Award was Gladiator. Facebook didn't exist. It was still 4 years out. iPod was just around the corner. The iPhone was still 4 or 5 years out. I had a hard time finding which was the most popular search engine. But in terms of like Alta Vista, Google was like 3 or 4 years old, was still up and coming. And it wasn't an Uber. If you need to go somewhere, like it was a taxi. And it would cost you $100 million to get to have your human genome sequenced. Today it's like $1,000, right? Or even, I think it was one company that's $600, but it didn't work out. So, and the most popular phone at the time was this Nokia. So things have changed. So that's like, well, how about 1980? So the kind of big phenomenon then was, I don't remember if you remember, I was alive in 1980, but Dallas, and the big thing was Who Shot JR? It was like this cliffhanger thing that, I lived in Sweden at the time, that came all over to Sweden. The movie was Kramer versus Kramer. There was no Facebook, so if you wanted to kind of share photographs and things, you got to have Kodak, right? You have Kodak moments. We all know what happened to Kodak. Right? Apple III was the product Apple released. They had just gone public. Apple III was a huge, huge, huge failure. This is supposed to be like the first business computer, didn't go anywhere. And if you wanted information, you better go over to a library and look at microfiche. I mean, how many people looked at microfiche here? Yeah. Yes. So it's very different. And yeah, good luck finding it. So you have to take a taxi and good luck trying to sequence your human genome. And then the state of the art for your mobile was Gordon Gekko's brick. So things change. So I have a lot of hope that neurostimulation, neuromodulation will really take off over the next couple of years, because I think that will really unlock our ability to modulate the brain in ways that will be very different. So thank you very much for listening. I hope this kind of spurs some ideas. I'd be very happy if any of you feel you can help solve my problem. Please find me, seek me out, and have a conversation.
You have 7 minutes for questions.
Okay, very good. Thank you.
Thanks very much. It was a great presentation. So beyond the work that you're doing in MS and autism spectrum disorder, it's really interesting that you're looking at bringing in a number of different technologies for both biometrics measurement as well as full loop, closed loop response, right? The VR and the like. What other etiologies are you looking at, and what other technologies are you looking to bring into Roche to bring those to market?
So, I mean, again, if you look at terms of therapies, it's largely still drugs and large molecules. And what we're doing in autism is kind of our pilot around digital therapeutics. But we're also looking at that for things for like multiple sclerosis, where we have the floodlight platform. Then in terms of new ways of diagnose kind of brain disease and things like that, maybe we basically look at everything. I think we have, and my colleagues, Chris, for example, who leads a lot of these programs. I think we have like 5 or 6 disease areas we're using EEG right now. We use basically digital in all of our disease areas in neuroscience. We think about, MEGs, so magnetic encephalography. I mean, we do MRI, CT. I mean, one of the beauties, there are downsides to work at Roche, and I think one of the beauties, we have a lot of resources. We literally spend $30 million a day on R&D. So that gives a lot of opportunity to look at a lot of different things. So for us, the challenge is less about resources. It's about finding where to spend them and being smart about it. And so that's kind of where we are right now. So diagnostic, we kind of look at all kinds of technology. Therapeutically, it's a little bit more. We're still a biotech company, so going into other like devices and digital therapeutics is breaking very, very much new ground for us. I hope that helped answer it.
Thanks. I'm just curious. There's a lot of, discussions and also evidence in terms of early intervention for things like ABA, speech therapy, and things in autism. Curious to know if you guys have done any longitudinal study in terms of efficacy of drugs, if they're applied earlier. In other words, can early diagnosis, along with early application of drugs, change the trajectory of things like autism?
So I believe so, I think so, but the person who really knows is right behind you. This is Kevin Sanders. He's the head of our neurobehavioral, so he knows everything about autism that's worth knowing, right? So, yeah.
So I think that the really critical factor here is, how do we detect change in the population in general? And then the younger kids, it can even be more difficult. In our molecule programs, we're going down to the age of 2. The FDA and the EMA really want us to cover that age range because they know that if we develop drugs, they'll be used in younger kids. But really the challenge for us now is how do we develop and improve our endpoints that we have to see how they work.
I mean, in general, I can't think of any disease where hasn't been shown that earlier intervention is more effective. I mean, pretty much all the diseases we looked at going earlier, sort of for oncology or the neurodegenerative diseases, early intervention. So I would think it would be true here as well. Walter? Yeah.
A lot of the innovation in this arena is occurring in the academic arenas or in the early stage startups. And so I'm wondering how does a company like Roche, what's the pathway for them to find those innovations and work with those earlier stage companies? Sometimes there's a bit of an impedance mismatch.
Yeah. So one is to come here would be one example. Yes. Yeah. Thank you. And if we do reach out, I mean, I used to be in venture capital, so I talk to my friends, colleagues. We have a Roche Venture Fund as well. Really, I mean, that's why I'm here. And the idea is that here are the things we want to solve. If you have something you think might address that, just reach out to us. But I do understand. I mean, the problem with Roche, I forgot, there's like 100,000 people. I even have an app on my phone that tells me where they work. Like I put in someone's name, I can tell them exactly where they work, etc. It's very helpful. It's hard to navigate an organization like that. And the reason we come here is even if we don't work on it, it's like say if you're doing something in oncology or somewhere else, I can point you in the right direction.
I'm interested in, with Parkinson's. My mother has Parkinson's, and I've just been kind of shocked by how unscientifically it seems that people do the drug dosage and timing of the dosage with respect, because there's some people who basically don't want to take any medication because they're afraid that it'll lose effectivity if they're young. And there's, again, like my mom had late onset, and the doctor at first said, well, no, just tell me if the medicine runs out, if you feel it running out, well, we can increase it. There's, you're just at the beginning, but get a very low dose. But it's all very, just kind of very hand wavy. There's absolutely
I agree with you, and I used to actually take care of a lot of patients with Parkinson's, and sorry to hear about your mom. I mean, it's the problem with the fluctuations of L-DOPA, right? Either you're frozen, or you take too much and you get dyskinesias from it, right? And timing that, and I mean, it's a fundamental problem. Our dosing, the way we dose drugs is very archaic, but trivial. Like we really need to figure a way of having a feedback loop around that, right? So for example, one idea we thought about is, with the sensors we developed, so we have a similar platform like an MS for Parkinson's disease as well that measures how well you move. Could you link that with a dosage? Now we don't have L-DOPA in our portfolio, but you can actually have L-DOPA. And actually I looked into that. Now the problem with L-DOPA, you can't really formulate it. You could basically create an artificial basal ganglia by having a pump with L-DOPA in it. You have some measurement of how your movement is, and based on that you dose it. So you would kind of like they're doing right now in diabetes, right, with artificial pancreas. And we need to get there at some point. I mean, one really promising with deep brain stimulation is that now they're going to start having a closed loop. So it used to be deep brain stimulation was just one way. but now they will also sense. You can start to create that neurofeedback loop. So I think a lot of things for neuro that I'm excited about is to create this biofeedback loop. And that's just not true for Parkinson's disease, but also others where you can, and actually that's what I was going to ask also the lady from Abbott, but the spinal cord stimulator, right? The more you can create a neurofeedback loop where people can adjust it depending how they're doing, and then you can also learn what wave patterns and stimulations actually work. You can start segmenting patient populations. So that's kind of what the vision we're working on. So that's probably a broader answer than you wanted, but I totally agree with you on Parkinson's. It's actually one of the fundamental challenge of treating it beyond slowing down the progression. We do have a drug that we're working on to try to slow down the progression, so hopefully that will pan out. Good. Good. Thank you. Thank you very much for your time. It's great to